Okay. Go ahead.
Did you know that according to the Centers for Disease Control and Prevention, birth defects affect 1 in every 33 babies born in the United States each year, making them the leading cause of all infant deaths?
This is a big problem, and one that scientists have been trying to solve since as early as the 1970s.
It is possible that they have created a system able to prevent birth defects and genetic diseases, but due to the cost, risks, and ethics involved, it has become an extremely controversial procedure.
We will be presenting the pros and cons of genetic modifications, and we will present to you, you can decide whether or not it is right for parents to genetically modify their children.
I'm Natalie Bubenheim.
And I'm Madison, and our presentation is over genetically modified children. Okay, so a brief history about genetic modification. The first genetic modification started in the late 1900s, and it was on tomatoes for farmers, actually. And the genetic modification allowed tomatoes to be increased in size and ripen later in the season, so it increased the farmers' crop yield. GMO food has rigorous guidelines before it even reaches the market, and the first GMO food to ever hit was salmon in 2015, and it was approved by the FDA. The salmon had increased hormone levels, so it reached full size in just 18 months rather than in 2 years. But however, the salmon came with hazardous health risk due to the increased hormone levels, but not only health risk did it come with, but also environmental risk factors. If the salmon were to be released into the environment, it could destroy the natural population.
A new method was used for genetic modification in 2012. This was the CRISPR method. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeat. The CRISPR method was actually modeled off of the immune system of bacteria. The CRISPR immune system works to protect the bacteria from viral attacks by targeting the virus's DNA and cutting it into short segments called spacers, which can then be inserted into the CRISPR sequence. These spacers then undergo transcription, where they are cut into short CRISPR RNAs, which can then guide bacterial molecular machinery in order to destroy the viral matter. Scientists have harnessed this technology in order to make precise changes in genes of different organisms, including human cells. They are able to remove or replace an existing gene, switch a gene on or off, or insert a new gene altogether. The CRISPR method is able to target multiple genes at once by finding the target DNA, locking on, and delivering an enzyme called the Cas9 in order to cut the DNA.
The first genetically modified child was born May 23rd in 2013. And it was born to a child, or to parents who were deemed infertile. According to Helpline, the baby was implanted in the mother's womb, and the doctors were able to control every gene that the child would ever have. So it helped with the mother's infertility. So there's a major bonus to genetic modification. It can help couples who are deemed infertile in some cases.
While the cost to edit a genome is extremely high, it has dropped exponentially in the last few years. According to the National Human Genome Research Institution, the cost to modify one gene in its early stages of research, it could cost up to $100 million. But through the development of the CRISPR-Cas9 system, that cost has gone down to under $5,000 per injection. This is because the CRISPR-Cas9 system is able to modify different genes using the same equipment just by flipping different guide RNA sequences into the editing system. Editing genomes in the human embryo using the CRISPR-Cas9 system could potentially prevent genetic diseases such as Alzheimer's, Down syndrome, spinal muscular dystrophy, and Huntington's disease. The technology can be used to inactivate or replace a mutant human gene in the zygote stage in order to prevent the genetic disease from affecting the individual. This process just involves injecting a target RNA, a Cas9 enzyme, and a substitute DNA sequence into one cell using a micropipette. After that one injection, every cell in the embryo will contain the edited DNA, and as the baby develops and matures, every organ in his body will contain the DNA, meaning it will not be affected from the disease that was edited. Since every organ in the individual's body contains the edited DNA, including the testes and the ovaries, that individual's offspring will also be immune from the genetic disease, meaning that an entire family or germline could be cured from a genetic disease just from one injection into one embryo.
Since genetic modification eliminates genetic diseases, it can also leave children open and more susceptible to regular viruses since their immune systems haven't all the way been built up. When undergoing the process of implantation, the zygote is implanted and there's room for human error. According to the New York Times, in one case, a mother who had dwarfism wanted a child who had dwarfism as well, but due to a mix-up in eggs, her baby would grow to be regular height. The medical procedure, there are several eggs involved, and every egg is manipulated until it's deemed superior, and then it's picked 1 out of 13 eggs. But this is the moral case of that doctors are holding a life in their hand and they're holding it back from becoming a baby. The zygote at that point is living. It's human cells who have the potential to become a fetus. So it's an ethical question of whether or not the process is okay.
While the CRISPR-Cas9 system does have the potential to make off-target cuts, which can result in unwanted mutations, that potential is very low. And according to an article called Genetic Engineering written by Michael Evans, that number has already dropped substantially since the beginning of research and from the development of the CRISPR-Cas9 system. Through more research and experimentation, that possibility could be dropped even lower. People argue that using the CRISPR-Cas9 system to edit human embryos is not 100% safe, and they're right, but no medical procedure is. There's always the possibility for errors or mistakes to be made, and it is impossible for a medical procedure to have an absolute 0% error rate. People are opposed to the research and experimentation of the CRISPR-Cas9 system and genetic modification as a whole because they say it is too dangerous. But the only way to make improvements to it and to make it safer is through research and experimentation. Furthermore, the CRISPR-Cas9 system can be used to edit the genetic makeup of a baby. This means that parents will be able to choose the outward appearance of their baby, including its gender, eye color, hair color, and more. They could also make changes to the personality, intelligence, and athletic ability of the baby. Since genetic modification includes discarding the defective genes in an embryo and keeping the healthy ones, a baby who has been genetically modified would grow up to be much healthier than one who was not. According to an article called What Are Designer Babies and What Do They Mean for the Future of Reproduction, written by Emma Feibeck, the overall life expectancy could be increased by up to 30 years. If the CRISPR-Cas9 system is more widely used in genetic modification with embryos, the overall average life expectancy of Americans could be greatly increased.
Since there's lack of guidelines for genetic modification, who's to tell them that you can't design a child with a genetic disease? There's also an ethical question of parents designing just to donate, where they design a child with a specific blood type so they can have it donate organ tissue, blood to another child. There's also an issue of social class. The procedure is expensive, and it could widen the gap between classes. Kids could get bullied in school because they don't have the superior traits that are in for that. But there's also an issue with tensions at home. Since parents can design their child to a T, the two parents may feel like they can have a control over their child, that if they are designed to be athletic, that they have to do sports, or if they're musically talented, that they should play all of the instruments.
In conclusion, I believe that parents should be allowed to genetically modify their offspring, mostly just due to the many benefits it has. If there is a procedure available in order to prevent children from having a genetic disease and preventing their future generations from also having that disease, that procedure should be utilized, and we should be helping to make people healthier. And also, just with parents who are infertile, they're really one of their only options for offspring is through adoption, and that child's not going to have any of their shared DNA. It's not going to pass down traits from their parents. So through designing the baby, the parents can give their offspring similar hair color or eye color to them, or certain personality traits that they would want their child to possess.
The ethical questions still being raised still raise valid red flags. There needs to be set guidelines before this is open to the public. We need set guidelines to follow to ensure that not only for our health, but also the future designs of the baby is safe.
And these are our references.
Okay. Thank you very much. You guys ever seen The Island?